Great. So, welcome everyone. My name is Marika McCabe. I am one of the scientific engagement managers with Protocols.io. And as I mentioned, for those of you who joined at the beginning, I'm based in our New York office. I have a background in biomedical sciences and am one of the scientists on the Protocols.io team. Today, I'll be presenting about our introduction to Protocols.io. And some quick housekeeping. As you heard, the recording started. We will be recording this webinar. You will receive a link to the recording after the webinar ends, probably tomorrow or the day after, along with these slides. Closed captioning should be enabled if you want to use captions, and I'll show you how to do that on the next slide. And then we'll have Q&A at the end, but you're welcome to add your questions during the call into the Q&A section. And we also will have a webinar feedback survey at the end, so you can give any feedback on my presentation, the webinar, and also if there's any topics that I didn't cover that you really would like for us to cover in future webinars. We do have multiple webinars throughout the year, which you can check out at protocols.io/webinars. This is our introduction, so I'll really be going over the basics of protocols.io and how you can get started using the platform. We have a tips and tricks webinar where we get into more advanced use of protocols.io, and then a getting credit webinar, which covers how to publish your protocol and some of our options for having your protocol peer-reviewed or associated with a manuscript. And you can request a demo and meet with us one-on-one if you haven't done that already and you have personal questions that you'd like to ask. You can enable captions at the bottom of your Zoom screen with this CC Showed Captions button here. When you click that, you'll be prompted to select your caption language. So if you need translations, you can change the language of translations. As some of you may already know, often sufficient information for repeating experiments is lacking. Methods are either lost or they're just not reported in sufficient detail. And this is common across a lot of different scientific areas of study. I have a biochemistry, biomedical background, but my favorite of these tweets is this one from a physicist because I experienced this myself, where you'll read a paper and in the method section it says, devices were fabricated or the experiment was done as previously described, and that references another paper, which references another paper, which references another paper, and now you're back a decade and it says devices were fabricated with conventional methods, and you might not even know what conventional methods were. So it's really difficult to know how to actually repeat that experiment. And sometimes it's really hard to even find the original reference because the methods are behind a paywall. So it references an article that you don't have access to anymore. This is really a challenge, and in 2025, a reproducibility project found that just by looking at methods and common methods in a single field, there was really low reproducibility. And this highlighted a weakness in how we not only produce our science, but how we document and communicate our science, and the fact that we really need to have open sharing of data and protocols. And what's really important is that it's not just about others who are reproducing or building on your work. A lot of the time, it's really you who needs the methods. And my favorite tweet is this one here by Karen Cranston, where you mostly collaborate yourself and with yourself, and me from 2 months ago never responds to emails. It's really true that as you're doing experiments, sometimes you think that something is really obvious and you don't write down what the method was or what the slight change that you made was. But later down the line, you might not remember what you did on a specific day for a specific experiment. So if you take the time to document your methods clearly while you're doing them, not only do you save yourself time in the future when you're repeating the experiment, but also that work is done for you when it comes time to share those methods openly and share those methods with others later. And open methods really fit into this frontier of open research. So a lot of the open research movement has focused on open access publications and open and fair data and code, but data sharing needs method sharing. If I gave you some chocolate chip cookies and that's the data I'm giving to you, and I asked you to reproduce that or even to interpret that data, you wouldn't necessarily be able to tell me how I got those cookies, what the ingredients are, whether it's something that someone with a nut allergy could eat because you don't know all of the intricate details of my method. So data can really only be interpreted in the context of how it was produced, and it's really important to start sharing your methods openly and making them available. Some recent policy recommendations around that include the ProMap from the European Commission and the UNESCO recommendation on open science. Protocols IO is built to make it easy to share method details before, during, and after publication, and we fit into this idea of really allowing you to be the best collaborator you can be with yourself, with your collaborators, and with any scientist who wants to build upon your work. There's two main sides to Protocols IO. We're one tool across the research cycle. The private side is our experimental workflow tool where you can document and track method tweaks. You can collaborate with your lab or global collaborators, and you can work to troubleshoot your protocol or take protocols that exist already and bring them onto protocols.io, which I'll talk about a bit later. We have over 96,400 protocols on the private side, and so these are accessible just to those researchers and anyone they choose to share them with. We also are an open method repository where you can share your method openly and receive a citable DOI. This allows you to then link the method to papers, get credit for any development work you've done for your methods, and then discover and reuse others' methods. So someone may have already created a method on Protocols.io that you can adapt for your own use and build upon. And there's over 27,100 public protocols on our open methods repository. We have a partnership with a lot of universities that offer premium access. A few are listed here, but if you go to protocols.io/for-institutions, there is a drop-down menu where you can check if your organization is listed and either create your Protocols.io account if you haven't created one yet, or upgrade your Protocols.io account to premium through your institution. We are also a Research for Life partner, so we provide free access to Protocols.io for any of the countries that are color coded in blue here, any of the Group A countries. So if you are potentially someone who qualifies for premium through Research for Life, I would encourage you to check the web page, and if you have any questions, to reach out and we can help you to gain that premium access. Protocols on Protocols.io are built for modern science. So moving away from a static document, you're able to update and improve protocols in real time. After you've published your protocol, as the science continues to adapt, you can add new versions. You could also create a fork of a protocol and modify either your own protocol or someone else's protocol for other model organisms, other cell types, other fields of study even. We have some advanced AI features, including assisted protocol troubleshooting and multilingual translations, allowing you to translate your private protocols to 36 languages and share them with any collaborators or even with yourself in these languages. Protocols.io is really built to be collaborative and to encourage you to work with others as you're working on science and methods development and using methods. So you can edit protocols at the same time as someone else. You can also add comments as shown here, where you could ask the author of a protocol a question and they can respond to you and clarify as you're troubleshooting or adapting the protocol for your own use. It's easy to find and follow the methods, and we allow for multimedia enablement so that if you have a protocol where maybe words don't do enough to explain how to properly do that protocol, you can add videos or images or GIFs or plate maps. And on the private side of the platform, we have granular permissions and selective sharing, ensuring that your private protocols are secure and are kept private until you're ready to share them with the broader scientific community. Protocols on Protocols.io also fit into this idea of modular publishing, where you can publish discrete research outputs that all come together into a paper. So a really good example here is this 2024 paper from Nature Communications, where the expression constructs were deposited in AdGene, the protocols were deposited on Protocols.io, and the molecular structure was deposited on Protein Data Bank. This ensures that all of these little components that come together to form a research article are deposited in a way that they can be fully utilized and fully understood by others, instead of just having your methods really reduced down to a few sentences in a methods section. You have that full step-by-step recipe-like protocol available. This also ensures that you get first author recognition. If you were really involved in the development of a method, you may appear on the paper. And in this case, Kelsey J. Ramirez is the fourth author on the paper, but one of her huge contributions to this paper was the development of this UHPLC MS-MS protocol, where she's listed as the first author. So separate research outputs are able to have distinct author lists and ensure that the authors are given the credit for how they contributed to the article. An additional benefit of modular publishing and of publishing your protocol separately from the paper is that there's a allowed method transparency, because all protocols published on Protocols.io are published open access. All protocols published by users on Protocols.io are published open access and with a CC BY license. That protocol is open, whereas the paper that cites the protocol or where the protocol was initially developed for may be behind a paywall and subscription based. And it's really nice because protocols remain transparent and tractable throughout the research publishing lifecycle. So we have this protocol for safe, affordable, and reproducible isolation of SARS-CoV-2 rainwater, and it was published originally in 2021 with a preprint in medRxiv. As the researchers continued to work on their science and continued to receive feedback on the protocol, they published a second version later that year with a few changes made, and that later was published was later cited in a corresponding PLOS One journal article. Later in October of 2022, the authors were able to even further adapt the protocol and publish version 3. And what that looks like is that anyone who accesses that PLOS article, when they click that DOI for the version that has now been updated, they can still view that older version, but they'll receive a lightbox pop-up that says there's a new version available, and they can view that newer version and remain up to date with how the protocol was adapted, and ensure that they're really seeing the complete version without needing to go back and have a formal editorial revision of the PLOS biology article. We also really encourage our researchers to share their methods on Protocols.io, in part because this can connect you with other researchers. This quote from Professor Luciano Martellado: 80% of recent collaborations have been a direct result of sharing our protocols publicly on Protocols.io. If you share your protocols publicly, others are aware of what you're able to do in your research and also are aware of new techniques that you may be developing and can reach out to you if they're interested in collaborating or working with you. And so this is kind of an overall summary and introduction to Protocols.io, and I hope that you'll appreciate from the previous slides and before we get into the live demo of the platform, that we're built to help you transition throughout the different stages of your research with Protocols.io. Whether you're coming on the public side of the platform to search and find protocols that are deposited there, or taking those existing protocols, whether they're someone else's from the platform or your own from your research, and optimizing and organizing them to the time that you're ready to share and publish, whether that's privately sharing with collaborators or with your own lab members, or publicly publishing and incorporating in a manuscript, or just publishing the protocol on its own for others to use. We'll now get into a live demo of Protocols.io. So this is the introductory webinar, so I'm going to cover a lot of introductory topics. For those of you who are really familiar with the platform, this might be a repeat of some of the things that you've already experienced. And after this webinar, I really encourage everyone to go on the platform, utilize it, and if you want to learn more about some of the more in-depth topics, sign up for the tips and tricks webinar that will be held later. So we'll cover workspaces, creating protocols, editing protocols, and then I'll briefly touch on running and publishing protocols and what a published protocol looks like. Close the PowerPoint. All right, so you should now see my Protocols.io screen. When you go on to protocols.io, you'll see this protocols.io/welcome. Um, you can search from our collection of reproducible methods, and I'm sure for any of you who pay attention to numbers, you'll notice that this 100,900 is higher than the 27,000 user-deposited protocols I mentioned. This includes Springer protocol content, so we have access also to Springer protocols if you are a subscriber to that. It is free to create an account on Protocols.io. And if you are creating an account through your institution where you have access to premium, or some grant funders as well give access to premium, then you'll have unrestricted access to creating new protocols on the platform, creating workspaces on the platform, putting files into your file manager. And if you're creating instead the Open Research account, you will be limited to only 2 files at a time, and you can have one private or one public workspace. So when we come into protocols.io, and I'm going to just put this in compact view, you will have the file manager. So the file manager is a place for you to store your files, find files that are shared with you, and share files with others. The file manager also is present in your workspace. So a workspace is a location where you can share protocols with your select collaborators, and they're stored privately in that file manager. But also, if you choose to have a public-facing workspace, such as this one, any of your public files will be shown to anyone who is viewing your workspace and interested in what you're working on. So in this case, the coronavirus method development community's public workspace, anyone can join if they want to, and that would allow them to view private files. But anyone who's just viewing the community can look at any of the public protocols, any of the discussion happening with the group, resources, and news. For your own workspace, it's also really nice to have access to this Impact tab. The Impact tab provides a summary of how many citations your protocols have had. So this is how many unique manuscripts have referenced your protocols. And you can scroll down and see for each protocol, the name of the protocol, the journal that it was cited in, and the complete publication, or excuse me, the complete citation of that manuscript publication. You can also view the total number of publication views. And so if you're running a project or a program where it's really helpful to have metrics around the impact of your science, having this impact tab allows you to view how your public protocols are being utilized by the scientific community and these citations and publication views. Excuse me. In the workspace, when you have created your workspace, you will be, let me refresh this for 1 second, since I changed my view settings. In the administration of the workspace, you will be the administrator of your workspace. This allows you to change any settings, invite members, manage the subscription if you have premium, provide file manager instructions to anyone. So these are instructions that tell anyone in that workspace how to properly use the workspace or a point of contact to use. And these can be accessed at the top right-hand corner eye here. And so you can see those file manager instructions. Those will also pop up for anyone who is first joining the workspace, and they have to acknowledge them before they can fully access all of the files. I'll go back to this administration on the left-hand side here. So if we go to members, this is where you're able to invite others to your workspace. You can have your workspace set to private, public, and then there's a few different options within that. So in this case, my workspace is completely private and people can only join my workspace if I invite them. I can invite my users by clicking this invite people and then copying a invitation link or inviting via email. And then once I click invite, they're able to join. If I realize at some point that someone has joined my workspace that should no longer be there or should not have been there in the first place, I can go ahead and remove them really easily. I'm also able to change any access settings. So in this case, my Protocols.io Marika McCabe account is the owner of the workspace, and my Springer Nature Marika McCabe account is a member of the workspace. But I could change that to be an admin. So admins are able to also manage workspace settings and members, or I could transfer ownership. So if you've had someone in your lab create a Protocols.io workspace for your lab and they're leaving, ideally the head of the lab would be the owner, but they can transfer ownership to whoever the appropriate person is. So it may be something that's uh, maintained and regulated by the lab manager. In that case, you might transfer the ownership. For those of you who have institutional premium, you can invite external members and they'll just be flagged that they're not part of your organization so that you know that someone is part of your workspace but not accessing premium, in this case through the Protocols.io organization. You're also typically limited to 10 external collaborators in a premium workspace. If we go to the settings here, this is where you can change your workspace profile picture, change any basic information or URL, add a workspace description, disclaimer, keywords that have to do with your workspace. So in this case, the coronavirus community has these interests that you can see here, their description that you can see here. And then if you have like a link to an external website, if you have a lab website, if you're a core facility and you want to link to your iLab, you can add that location affiliation. In security and privacy, this is where you're able to control whether your workspace is public or private. So as I mentioned, mine is private, internal only, and I have to invite anyone to join. They can't just join by finding this workspace. However, if the workspace is switched to being public, it could remain by invitation only. So anyone can view this workspace and can view my public files, but they can't join the workspace and they can't view my private files. I'm going to just go ahead and show you what that looks like for 1 second. So if I copy this workspace URL and I open an incognito mode, there we go. Now that this workspace is public, I'm able to view it. I'm able to come in and view the public coffee prep protocol. That's a protocol that I published previously from the workspace. I'm able to view any members. If I was signed into my account, I would be able to view any members who are public. But I'm not able to view any of the private information, and I'm not able to join that workspace. If instead I wanted to switch that to open to all, anyone could click join and join the workspace, or by request, users are able to then come view the workspace. This might not work since I didn't log in. There we go, and request to join. And of course, they would need to be signed up to Protocols.io or logged into Protocols.io to join the workspace. I'll go ahead and turn my workspace back to private. And I'll also note for those of you who are part of a premium institution, you can make the workspace visible to your organization. So when you check that, that means that anyone who is part of your organization will be prompted that this workspace exists and they could potentially join it. You can also add contact information and manage your notifications if you have a technical support contact information. And then you can set up your own notifications. If you want to know via email immediately when someone has made a comment, you can do that. If you want to know when a protocol is published, you can turn that on, and you can really change any of that information here. And then again, the membership and access gives you that invite link. You could allow other members to invite people to the workspace, and you can manage access here. This allows you to change what members are allowed to do and what admins are allowed to do. Again, a lot of this is only for premium. So if you are in a open research account, you might go in and not be able to change these settings. But just walking you through, if you're part of our, if you're with a premium account, you are able to decide whether or not your members can share files outside of your workspace. This would be creating a link for others to view, whether they can share files with the entire organization. This is also managed at the organizational level. So if you don't see that as an option, it might not be turned on for your organization. Allow publishing. So if you are a PI or a lab manager and you do not want students in the lab to publish protocols without your approval, you might want to turn that off so only admins can publish that protocol or any protocols in the workspace. Um, Allow moving files, removal of files, and the others are relatively straightforward. And then, of course, make sure to save your changes once you've made them. Uh, so now when we look on the left-hand side here, we have administration, which I walked through a lot of. You also can have different categories. So if you have a lot of public protocols that you want to highlight, in this case, I only have one, so it's a little complicated to show, but I can create a category. So my coffee prep, I can select that and say it's in this cooking category. I could create a new category that's research experiments, and this would be a way to categorize my protocols. And now when anyone is viewing my public protocols, they could sort the protocols to only see ones that are in that cooking category, to only see ones that are in the research experiments category, which I don't currently have any published. And that allows it so that your members, when they're viewing the workspace profile, can more easily navigate between the different types of protocols. Um, I mentioned you can also update the file manager instructions. As an admin, you can also remove any published protocols that you don't want associated with your workspace. So if someone has associated a protocol with your workspace and you don't actually want the workspace linked, you could remove that. Um, and um, you can send a notification to all members. For those of you with Premium, you can view your history of translations for private protocols. Control signing, which is a feature that allows you to lock a protocol so that it's uneditable, but it's not public, it's still private. That is also one of our premium features. So if you are on a premium plan, you'll see that as an option, and you can see which protocols were signed there. Um, and then you can view usage statistics as well of which of your users are using a lot of storage in the workspace, how many private protocols they have, what folders they have, what public protocols they have. And that allows you to really understand the users in your workspace. All right. So I went through how to invite people, the settings of file access, public versus private protocol sharing, or excuse me, public versus private workspaces. And so now let's really get into creating protocols. There's a few different ways to create a protocol on protocols.io, and I'll show a slide summarizing this later. But the very first way that I want to talk about is forking a public protocol. So if you're on protocols.io and you find a protocol that you really like and you want to work with, you can create a fork of that protocol. So in this case, I'm going to pull up this pipetting v4 protocol. This is version 4 of their protocol, and I can click copy/fork. Now, after you've clicked copy fork, if you're the owner of the protocol, you can make a copy. So if you want to create a copy of the protocol, you'll lose the link between the two, but this would allow you to keep maybe the author list the same, the disclaimer the same, some of the other information the same, but they will not be linked. Anyone else, you can click make a fork. You can change the name of the fork. So I could say pipetting with a multi-channel. You could change the language. Public protocols are all translated into English, German, French, Japanese, and Chinese. So public protocols, no matter who you are, you'll be able to view in these languages, and you could change the language in the fork. And then click Select Folder, choose which organization and which workspace you want to create the fork in. And then I'll go ahead and put this protocol into my folder called folder and click continue. The fork of the protocol, when it's ready, will show this pop-up, and then you'll want to click Edit New Fork, and you can come in now and make any changes to the protocol that you wish to make. I'm going to pause there and we'll come back to how to make changes to the protocol in a minute. But that's the very first way that you could create a protocol on Protocols.io is by forking someone else's protocol. I'll also mention that depending on how much change you're making to the protocol, you'll want to really be aware of the fact that in the descriptions tab, you'll have the original protocol authors listed, and you might make sufficient changes that some of those protocols should be, or excuse me, some of those authors maybe should be removed. You should be added as an author yourself. You may make enough changes to change the keywords or the external link in this case, I would definitely remove because I'm not part of their program. Um, and I probably would consider changing the image and checking the image attribution as well. When you've made a fork of a protocol, it will always be linked to the original version. So in this case, this COV2 protocol shows that it was forked from an Ebola virus sequencing protocol, and that ensures that the person whose work you're building off of gets credit for the fact that they kind of set a foundation for you. People can also compare the versions. So you can come over to the three dots, click compare, and compare the forked protocol with the original protocol and see just how many changes were made between the two protocols, what remained the same. So here you can see this was all in the Ebola sequence protocol, but was not in the COVID protocol, and really understand how much change was made between the original and your version. The next way that you can create a protocol on Protocols.io is with our AI importer. So these are, I should take a step back and clarify, these are ways to create a protocol from existing without building from scratch. So our protocol importer is a really helpful way to convert protocols from any format, whether that's a typed Word doc, a PDF, a JPEG where you've written by hand your protocol into your lab notebook and taken a picture of it. You are able to click that file once you've dragged and dropped it into your file manager, and then click this create a new protocol from this file. I've clicked the wrong thing on the right hand side there and click convert. You also can click new, um, import a protocol, and here you'll see the different file types, and you can upload your file by dragging and dropping it into the file manager and clicking convert. On the left-hand side, there will be a pop-up that appears that tracks the protocol progression. And just for the sake of time, I'm going to show you from my IHC protocol that was a Word document. It was converted into this IHC step-by-step protocol IO format document. And you're able to view that the different sections are there. I edited this slightly so that there were differences with step cases that we can get into later. Antigen retrieval, blocking, everything was present. You'll want to note when the conversion happens, if this is a private protocol you've been using in your lab, there's a chance you might have information like username and password that you don't want to share. In a private side of Protocols.io, you should leave it there. It's helpful for people to follow. When you go to publish, make sure to remove that private information. The other way that you can utilize an existing protocol on, or take an existing protocol and put it onto Protocols.io is with our protocol import service. So this is offered to our premium members at no cost, and for anyone else, it starts at $50. When you submit the protocol, we will assess it and determine what the actual cost would be before we charge you or request any payment. So you'll drag and drop your protocol or provide a link to it, add any notes, and submit it to us. We'll then go through and look at your protocol, enter it, double check that it compares well to the document you sent us, and then send it to you for review. If you want any changes, we can make changes. If it's good to go, we'll transfer ownership to you, and then you can edit it further. At that point, you can either keep the protocol privately for yourself or you can publish it on the platform for others. And then if you're starting entirely from scratch on Protocols.io, the way that you'll do that is by coming to this new and clicking protocol. You'll then pick whichever folder that you want to put the protocol in and can click continue. And you'll be met with this blank document template so that you can really begin anywhere. The first thing that I recommend for people is to add sections. Sections allow you to have a table of contents later. So when people are viewing the protocol, or even when you're viewing the protocol, it's a lot easier to follow. So in this case, if I have section 1 and then I'll insert section 2, you can see on the right-hand side here this table of contents. And when someone's viewing the protocol also, they can navigate between the table of contents. Of course, if you only have 2 empty steps, first of all, we wouldn't publish it on the site, but second of all, people would not really easily navigate or need to easily navigate with the table of contents. But this is really helpful when you have like a 100-step protocol. As you begin to edit, so section 1, we can say step 1. You can insert additional steps. If a step really needs to be broken up, you don't want a huge block of text. You want maybe 1 or 2 lines per step so that a reader can easily follow it. You might want to add in sub-steps. So here you would click Insert Sub-Step. And then you could just add in a few details. So you can add sub-step 2.1, another sub-step, and if at some point you insert a sub-step, but you actually wanted it to be a step, you just select that and click Convert to Step. One other thing that I'll note is that if you've already typed your protocol somewhere else because you didn't know about Protocols.io, you don't want to use the AI importer or you don't want to use the protocol entry service. One other way that you can add the details is click Insert Steps from Text, paste the text into this paste the text into this box here. So I'm just going to quickly type step, step, step. And then you can select either each number as a step, each line as a step, or mark down the text yourself. And then this will convert the protocol for you, and you can click Add to Protocol. You could do that for the whole protocol, or if there's a few steps that you had typed somewhere else and you just want to add those in. And then you can select multiple steps with shift click and delete them if you don't want them. If at any point you realize that you've made a mistake, you can undo. And for our premium license users, you can click history and view a complete version history. For those of you on an open research account, it is limited to the last 10 changes. So if you're on premium, if I made a change today and then in a few weeks after editing this protocol a few times, I wanted to go back and see what I had done today, I would be able to see that. But for our open research account, you will be limited to just the 10. Components are a really useful way to make your protocol not only more reproducible, but also runnable. We have a lot of components here, and our tips and tricks advanced user webinar gets into these in a lot more detail. But the ones that I want to highlight for you today are the amount component, which makes your protocol runnable. So if you put an amount such as 10 microliter, and maybe we'll go back here and put 10 microliter of water. That component can be scaled later, which I'll show in the run feature. For reproducibility, saying water is not really enough for someone to be able to reproduce your protocol. There's a big difference between the water from my Brita filter, the water from a Millicue, the water from distilled water that you purchase from a company. So we really encourage you to use the reagent component. You can search for reagent. So here I'm actually going to put distilled water. And this will go ahead and search reagents. You can find the exact company, the catalog number. We do have a partnership with Site AB, so we have a lot of their listed reagents. So in this case, let's say I used this Thermo Fisher one. I can add that in, and that's a much more specific way so that people know exactly which reagent I used. If the reagent isn't present in the list, you can click Create New Reagent, Buffer 1. If it's something that you make yourself in your lab, you can say that it is homemade. and then it would not have a catalog number, URL, CAS number. But if it is something that's available for purchase, we really encourage you to ensure that all of these details are added and include the RRID. So let's go ahead and create that buffer one. Now, when someone views my protocol, they can click that reagent and they can see if there are other protocols that use that reagent. In this case, there's no public protocols that use that reagent, but I'll show you an example of one that is used. Actually, um, so this NEB kit, you can see its information, the store URL created by NEB, and then it was used in 6 protocols. And if I click one of those protocols, I can scroll down. They have multiple reagents there, so I can view how they've used them. And the NEB kit is listed there. And then if any changes are made to how they use a protocol, ideally, or excuse me, a reagent, ideally you would put that into your information as well. Going back to components, so the amount component is really helpful for running and using a protocol. Reagents, software, equipment also are really important for reproducibility. So you could have a Zeiss microscope that is really important that people know the exact name, type, and brand of the microscope. And then there's a lot of other components that you can add to enhance your protocol. And again, I encourage you to attend our tips and tricks webinar to go through those. So I mentioned a couple times now the runnability and why it's important to have this amount component for runnability. So if you have a protocol like my immunohistochemistry protocol, where you have put in a lot of different components, and included, all of your reagents, temperature, timers. So this is just also an important one. Over here, duration, you can add a timer. This allows users to, such as yourself, to run the protocol. When you click run, you'll be able to create a run record of the protocol. You can start that run. Excuse me one second. You can start that run of the protocol. If you have an experiment ID that's a specific experiment ID, add that in there. And then this is where components really come in handy. So you can start here and utilize the component timer and actually start and run a timer. You can mark steps as done when they're completed. If you make any changes to the protocol, you can click edit and you can directly change the text of the protocol. Um, Here, I probably wouldn't make any changes, but I could put incubate all steps, all sections in the following and save that change. And then for the amount component, we allow for scaling. So this protocol is written for one slide tub, which held about 10 slides. If I wanted to do this immunohistochemistry with 30 slides, I would come to the top here, scale protocol, and scale by a factor of 3. One protocol for 10 slides, 3 times for 30. And you can see all of the steps that have volumes are now scaled. So this is a really useful way for you to keep track of what you did on which days, at what time. And if at any point you need to pause the protocol, you can come back and find it in your run records or whatever file you put it in, and you can resume the protocol. You also are able to share the protocol with others. So if you start a protocol and someone else needs to finish it, they can pick up the run record where you left off. And when you're finished, you will have a complete run record, which is uneditable except for comments, which you could add a comment if you wanted to. And you can export that as a PDF to your computer to print out and put in your lab notebook, or if you utilize a electronic lab notebook to one of our integration partners. I'll mention quickly now, the integration partners. So here, if you select export from any of them and you see PDF, you'll see that export a PDF to your computer. And then for any of you who are new to Protocols.io, you won't see any of these options. You'll need to click Add Apps. It will take 1 second to load, and then you can turn on and connect any of these partner integrations, and you can send your protocols from Protocols.io to these electronic lab notebooks or drives. You also can pull in protocols for SciShore. We have a bi-directional integration that's really helpful. So if you have your protocols are already there, you can pull them over to Protocols.io. All right. So as I mentioned, components we get into a lot more in the other webinar. So that was just kind of a high-level overview of components and of running protocols. And the very last thing I want to touch on today is publishing protocols. When it's time for you to publish your protocol, you have a few options. And I'll show you a few examples here. So an unpublished protocol will always appear as a yellow private protocol. It's unpublished. As I mentioned, for those of you with premium, you do have the ability to sign and lock protocols. So that will be a red protocol. If I view that protocol, you can see that it is signed at the top. It is private. No one can see it unless I've sent it to them, but it's uneditable. I cannot edit this protocol. A protocol with a reserved DOI will show up in purple. A reserved DOI is really helpful if you're preparing a manuscript and you have a protocol with methods that are integral to that manuscript, but you're not ready to make the protocol public just yet. You really want to time it at the same time as the manuscript. In that case, you'll go ahead and reserve the DOI. And now when you view that protocol, you'll see that it's still private. It has that private icon, but you have a reserved DOI. If you click this info here, you'll see the DOI that can be put into your manuscript and a private link that you'll want to share with reviewers to make sure reviewers can actually view and access your protocol. You can also find that in the metadata tab, the protocol citation, and you can use this cite this button as well to get the protocol citation. But with the reserved DOI, again, make sure that you add that private link for reviewers for them to access the protocol. And then when a protocol is completely public, as I mentioned previously, it will now be translated into English and 5 other languages for anyone to view. That protocol will now appear as a public protocol, and people can export the protocol, run the protocol, or make a fork of the protocol. You're able to see how many people said the protocol works for them. So once someone runs the protocol, if it worked for them, they can give it a thumbs up. People can bookmark the protocol to save it for themselves for later, and then you can track the views at the top here. If someone uses your protocol or you use your own protocol, you can click that handy Cite This button and either copy or download the protocol citation. If you download it, that's a bib file that can go into your citation manager. And then you can view the protocol and view details like how many people have made forks of the protocol. If it is your protocol, you'll see those details. If it's someone else's, you'll only see public forks. You can access troubleshooting. So if you have a premium Protocols.io account, you can type in a question here to troubleshoot the protocol. If something's not working and you want to kind of get a little AI generated feedback to help you figure that out. You can view the metadata information about the protocol, including the license, ownership, if it's a parent protocol in anything, or if it's in any parent protocols, and then metrics, so you can see how often people are viewing your protocol, or if your protocol is being cited, which let me pull up this protocol, you can go into the metrics tab and view the times and list of citations of that specific protocol. As your science adapts, you might want to create a new version of the protocol. So when you're viewing the protocol, you can click the 3 dots and click new version, or if you're in your file manager, you can click New Version over here, and you can make any changes that need to be made in order to better help others to understand and follow your protocol. And I'll also note that you can add comments. So if you haven't made substantial changes, but you realize that, in this case, I put NB distilled water preferred, tap OK. People might not know that NB means nota bene, and that just means it's a note. So NB means it's a note. I could add that as a comment and put it there for others to see without needing to publish a whole new version of the protocol when it's something minor like that. I also could find someone else's protocol and add comments in that case here. And then when you're viewing a public protocol that has a lot of comments, let's go to version 3 of this protocol, that has a lot of comments like this one, you can click that right-hand comments and see an AI-generated summary of all of the comments, and then also navigate to see if others are asking your question by searching the comments and seeing some of the notes at the protocol-wide level or the step-wide level. All right, so I have tried to cover everything. I just realized the one thing I didn't mention is how to actually publish the protocol. So when you are in your file manager and you're ready to publish your protocol, you can click this publish button here or reserve DOI button here. Or if you're viewing your unpublished protocol, at the top left here, you can click Publish or Reserve DOI, and both of these will take you through a 6-step process to ensure your protocol is complete, all of the author information is available, and that you're actually ready to publish. All right, so I have covered all of the topics of our demo. I know that there is a lot to cover, so if there is anything that we didn't get to, we'll try to get to that in the Q&A. I'll mention also we aren't just wet lab methods. I am a biochemist by training, so I show a lot of wet lab methods, but we do have methods across all sorts of fields as shown here. And we have this protocol of examples of protocols across fields. If you want some, I'm putting that in the chat, there we go. If you want some encouragement of what other protocols look like. And I covered that it's really easy, or we try to make it really easy to get your existing methods on Protocols.io or modify a published protocol, and you can really fork an existing protocol, use our entry service, import from text, or start from scratch. And just when you leave today, I hope that you'll think about one protocol that's really important to the work that you do and how you can get it onto the platform. We are encouraged by a lot of publishers, a lot of funders, and a lot of institutions. And we are publisher agnostic. So if you want to publish a manuscript or you've already published a manuscript in a journal that is not owned by Springer Nature, that does not limit you from putting your protocol on Protocols.io. And as I mentioned, we have a lot of topics to cover, so I encourage you to sign up for our other webinars, to visit our help center, protocols.io/help, to watch some of our tutorials, submit feedback, or email us if you have any issues or want additional training. And we also record all of the webinars, so you can watch past recorded webinars there. I encourage everyone to fill out the webinar feedback form. That really helps me to know what I should have spent more time, what I should have spent less time on, or what topics would have really helped for us to cover either in the intro webinar or in a completely separate webinar. And feel free to send me an email, marika.mccabe@protocols.io, or email info@protocols.io. That is our entire team and is covered across a lot more time zones and not limited to if I'm ever taking a vacation day. So that's probably your best bet for assistance. Does anyone have any questions? I am going to stop recording in case anyone wants. Oh, let me start with this question first. So someone anonymous asked, when you fork a protocol, can the original author see who forked their protocol? So when you fork the protocol, if you've kept the protocol private, the original author will not see that you have forked and created a private protocol. But once you have published the protocol, they would be able to see your public fork. Okay, I'm going to stop recording now, but I'll still be here.